Good morning, students. Bio 102, nervous system. We're going to have a big discussion on not just the nervous system, but everything that is connected to the nervous system or gives information to the central nervous system. And those would be the sensory organs as well as the nervous system itself. So this is going to be a nice, long, big trip. So let's get started. Well, why do we actually have to have a nervous system? And the reason is communication. Many animals have a complex nervous system, and it consists of several parts. There's generally a central nervous system, or CNS, where integration of information takes place. This includes the brain and a nerve cord. And then there's usually a peripheral nervous system, or a PNS, which carries the information into and out of the CNS. So that's two big ones. And then, of course, the neurons of the PNS, when bundled together, form nerves. So we have all these parts that make a complex nervous system, which serves to communicate between all the organs of the animals so that it can be a harmonious coordination and the animal can benefit from all the information that it's been given to by the environment, so it can live productively. The basic unit of communication in the nervous system is a neuron. So we're going to look at the cell itself that comprises all these parts of the nervous system. Neurons are nerve cells that transfer information within the body. They have a specialized function, and that's all they do. That's all they're used for. Neurons use two types of signals to communicate. One is electrical signals, and those are for long distance. And then there are chemical signals, and those are for short distance. We already looked at the chemical signals previously. We looked at nerves communicating with the next nerve at a synapse, and we saw little neurotransmitter molecules falling out of the foot of the axon of the nerve and then onto the head of the next nerve. And we did actually see that in the previous lecture, but those would be the chemical signals, and those are in a very short distance. But we also have electrical signals, and those are fascinating, and we're going to actually look at how do nerves communicate by electricity. Processing of information takes place in simple clusters of neurons called ganglia, or a more complex organization of neurons called a brain. So all these signals are actually integrated in small areas called ganglia, where the information that they just received is processed. And what does that mean? That means that the cells are talking to each other and saying, hey, I got this part. Hey, did you get that part? And so then it becomes more coherent of a signal, and then it's sent on to the brain. And the brain says, I see it. And then we do the appropriate action that is required. Here is a picture of electrical and chemical signals. So remember, the chemical signals are just at the foot or the axon of a neuron, and they're just the little chemicals that come out in a very short distance, and they are received in receptors of the receiving cell, which is usually another neuron. And then there are electrical signals, and those are signals that jump from part of the neuron to the next part, and they are things that we're going to actually look at in big detail. So what is the definition of a neuron? A neuron is an electrical excitable cell that processes and transmits information by chemical and electrical signals. They are the core components of the nervous system and do not divide. So when we looked at embryology, we saw that the notochord was formed and the nervous system was formed from the ectoderm layer, and the fetus usually has a lot of nerves that are formed in the beginning, and then suddenly they're actually taken away by apoptosis, because the finished product doesn't need that many. So then when the fetus becomes an individual after it's born, they come with all these neurons. They too are trimmed off. Many of them are trimmed off as the baby matures, and but ultimately you end up with a certain set. But what this really means is what you're born with is what you get, and what is trimmed off is trimmed off. Too bad, so sad. But neurons just don't divide. So these are the cells that stay in S0 phase. When we looked at the cell cycle and we looked at how some cells go into the S phase and then M phase, and which would be the dividing phase, but some cells actually just stay in the resting phase, and that's all they do. They just don't divide. So neurons are cells that do not divide, and this is useful, but it is also important to know that once it's gone, it's gone. What is a neuron? It has a cell body, it has an axon, and it has dendrites. And let me show you a picture of this. Well, it'll come in a minute. Most of the organelles of the neuron are actually in a cell body. Most neurons have dendrites, which are highly branched extensions that receive signals from other neurons. And then a neuron typically has one big dendrite, which is called an axon. It's a much larger extension that transmits signals to other cells at synapses. There is a cone-shaped base of an axon that's at the base of the axon and the neuron, its cell body itself, and that's called an axon hillock, and that's where all the information, the electrical information collects until it is ready to be transmitted. So here we have a picture of this, what we just talked about. So this is the cell body itself. That's the cell itself, all right? So just a regular old cell with a nucleus and all the other organelles that cells usually have. But the difference is that neurons have these dendrites, which are extensions of the cytoplasm, and they're used to talk to neighboring neurons. And then neurons also have one very long dendrite, which is generally called a foot, and it talks to the next neuron. So like here, you would see the foot attached to the head of the next neuron, and so it's talking to the next neuron. At the synapse, which is right here, this is where the neuron cell body has generated all these neurotransmitters, which are going to go down, 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 and into the next neuron. So these are neurotransmitters. We looked at them in hormones as well, and we saw what they do. So this shows you briefly the structure of the nervous system, which is made of repeating units of these neurons. So let's talk about the neuron structure and function. The synaptic terminal of one axon passes information across the synapse in the form of chemical messengers called neurotransmitters. A synapse is a junction between an axon and another cell. So that's the definition of a synapse. Information is transmitted from a presynaptic cell, which would be a neuron, to a postsynaptic cell, which is usually another neuron, but it could be a muscle or it could be a gland. So we saw that sometimes there are endocrine glands that are under the influence of their neurohormones and neurotransmitters, and so they have to actually end up receiving the signal. Most neurons are nourished or insulated by cells called glia or glial cells. So remember, these cells never ever divide, so what you have is what you keep for the rest of your life. So these cells have to be maintained, and they have their special helper cells called glia, which nourish these delicate neurons throughout their lives. So here's a structure of a typical neuron, and this time it's not attached to another neuron, so you can actually see what is going on. So we still have the cell body, and we still have the cell nucleus, and we still have dendrites, but now we're looking at the foot or the axon of the neuron. What you actually see are these areas called, well, they're myelin sheaths. They're actually a single cell, and I'll show you a picture in a moment, which is wound around and around and around and around the axon, and it serves as insulation because it's fat. However, in between that, there is always a little gap, and that is where, so this is the hillock of the neuron where the the electrical information collects, and then it's going to jump from node of Ranvier to the next node of Ranvier to the next node of Ranvier and down, because electrical signals can then move much faster than chemical signals. And these areas that don't have myelin sheaths are deliberately left bare so the electrical signal can pass, because otherwise it would just be, the foot would be covered by fat, and fat does not conduct electricity. Sorry. So these myelin sheaths are produced by cells called Schwann cells, and like I said, I'll show you a picture of a Schwann cell in a moment. A neuron has only one axon, which can be as long as 1 meter, and you might wonder, why is that? Well, remember, the neurons are part of the central nervous system or the peripheral nervous system, and you have legs. So even if the last neuron is inside your spinal cord, the foot of that neuron has to go all the way to your toes, and that, depending on your height, it could be a whole meter long. So and the axons can be very, very long. Neurons communicate with each other at synapses by discharging electrical signals. All neurons are electrically excitable, and this is very important, and we're going to look at the fascinating way that neurons talk to each other by electricity. What exactly is electricity? It's just a current. And how do you get a current? Well, you have positive and negative ions, and that is how you send a current. So neurons are talking in ions, and that is what generates the current. Neurons maintain voltage gradients across their membranes with ion pumps, which generate concentration differences of ions like sodium, potassium, chloride, and calcium. These are the 4 ions that help us talk in electricity in our body cells. So this is awfully cool, and this is what too much of one ion will generate a voltage gradient. So instead of a concentration gradient, now it's a voltage gradient because it's actually a charge. Every cell has a resting potential or a voltage. Remember, a voltage is a difference in electrical charge. Every cell has a voltage across its plasma membrane, and that is called a membrane potential. The resting potential is the membrane potential of a neuron, which is not sending signals. So when a neuron is at rest, it has a certain voltage, and that voltage is called the resting potential. Changes in membrane potential act as signals, transmitting and processing information. In a mammalian neuron at resting potential, the concentration of potassium is highest inside the cell. Potassium is highest inside the cell at resting potential, while the concentration of sodium is highest outside the cell. So we have sodium-potassium pumps, which are in the membrane, and these use the energy of ATP to maintain these potassium and sodium gradients across the plasma membrane. So we want to always keep a lot of potassium inside the cell and less sodium inside the cell, and not have it go to equilibrium, because then we can't maintain a voltage gradient, and we want to do that to be electrically excitable. So we have these pumps which are constantly using ATP to keep that voltage gradient. These concentration gradients represent chemical potential energy. Here are the ion concentrations inside and outside mammalian neurons. So let's take a look and let's see. So when I said potassium is kept inside, look at the enormous number, 140 millimolar. And look at outside, it's only 5 millimolar. So that's a lot of energy being spent in active transport, keeping that much concentration of potassium inside the cell. Because it really should be balancing what is outside the cell. And then you look at sodium, and it's almost the reverse. There's a very little sodium inside the cell, but there's a ton of it outside the cell. And you look at chloride, which is a negative ion, and it's the same as sodium. And then you have large anions like proteins. Proteins are always negatively charged, and they usually have a very, very big negative charge, and so therefore they will add up the charges so that they're balanced. But again, you won't have, it's not the same ions on both sides of the membrane. The opening of ion channels in the plasma membrane converts chemical potential energy to electrical energy. So that's what happens. So we have these chemical potential energy in the form of ions inside the cell, and when we open up the ion channels, then that chemical potential energy changes into electrical potential energy. A neuron at resting potential contains many open potassium channels and fewer open sodium channels. And that makes sense because we want to keep a lot of potassium inside, and we want to keep a very little sodium inside. So it's not going to have a lot of sodium channels open so sodium can rush on in. That's making more work for itself. So it's not going to do that. And potassium is going to diffuse out of the cell, and the resulting buildup of a negative charge within the neuron is the major source of the membrane potential. And we're going to look at membrane potentials and how they actually work. So here is a picture which you can see of sodium and potassium ions and the sodium-potassium pumps and the potassium channel and the sodium channel, and they're all color-coded. And so what you see is here is a neuron, and we're just going to look at this part of the neuron, which is enlarged over here. And what you see is the outside of the cell, and there are a lot of round ions which represent sodium, but not so many diamonds which represent potassium. But if you look inside the cell, there are a lot of diamonds which are really potassium, and not that many sodium ions which are little round balls. And then you see sodium-potassium, one sodium-potassium pump, and you see potassium channels, but not that many sodium channels. And that is because we're going to not want a lot of sodium coming in, so we have very little channels for it to come on in. The resting potential can be thought of as a membrane that separates two chambers. The concentration of potassium chloride is higher in the inner chamber and lower in the outer chamber. The ion potassium is going to diffuse down its concentration gradient to the outer chamber. So that's just going to happen. And that's Fick's law, and we saw that in diffusion. That's just what happens. And because that happens, a negative charge of chloride builds up in the inner chamber. At equilibrium, both the electrical and the chemical gradients are balanced. We also have, therefore, an equilibrium potential, and it makes sense because there's still a voltage at equilibrium. The equilibrium potential, which is abbreviated as capital E subscript ion, is the membrane potential for a particular ion at equilibrium and can be calculated using the Nernst equation. So this ion could be any ion. And so this is a general equation, and it shows you the equilibrium potential. How do you calculate it? So it's 62 millivolts times the log of the concentration of the ion outside divided by the concentration of the ion inside. And remember, the square brackets always denote concentration. The equilibrium potential of potassium has already been calculated, and that would be actually written as, therefore, E sub K, and that's negative, while the equilibrium potential of potassium is positive. And here we actually see that. And so we have an inner chamber and then the outer chamber. So this is simulation of a cell. We have these nice channels. Yeah, these are the potassium channels. And inside we have a lot of chloride ions and a lot of potassium ions. So it's potassium chloride, and it's 140 millimolar. outside in the interstitial fluid, it's only 5 millimolar, and you want to keep that. And so that's how you would actually calculate the equilibrium potential of potassium, 62 millivolts. It's the same equation we just looked at, and times the log of 5 millimolar, so that would be the outside, divided by the inside, which is 140 millimolar, and you get to negative 90 millivolts. All right, when you divide it out, that's what you would get. And then when we look at sodium, how do we get that potential, the equilibrium potential for sodium? Well, we're looking at the inner chamber, so this would be the inside of the cell. And inside of the cell, there's only 15 millimolar sodium chloride. And you see some chloride and some sodium. But outside the cell, you actually see a lot of chloride, sodium chloride, both chloride and sodium. So we're going to just use the equation again. So again, it's 62 millivolts divided by log of, excuse me, the outside, which is 150 millimolar, divided by 15 millimolar, which is the inside concentration of the ion, to get positive 62 millivolts of equilibrium potential for sodium. In a resting neuron, the currents of potassium and sodium are equal and opposite, and the resting potential across the membrane remains steady. Changes in membrane potential occur because neurons contain gated ion channels that open or close in response to stimuli. So then there is the concept. We talked about equilibrium potential, but now we're going to talk about hyperpolarization. When these gated channels, the potassium gated channels open, potassium ions are going to diffuse on out. That is going to make the inside of the cell more negative. It's already negative. Now it's going to get more negative. You don't really want it to be more negative, but it is going to get more negative. And this is called hyperpolarization, which means, hyper means more, an increase in the magnitude of the membrane potential. So we're going to look at the voltage gated ion channel. Yeah. So we have the gate closed over here. No ions are flowing across the membrane. There are ions outside, there are ions inside, so there's nothing happening. But when the gate's open, the ions are going to move down in the concentration gradient, down the gradient. So all these ions are going to flow on in because inside is not that much. So it's going to go equalize it. So that's going to change the membrane potential. And then you have hyperpolarization. So we can have graded hyperpolarizations, which are produced by two stimuli, and that increase the membrane permeability to hyperpolarization of potassium. So if this is the membrane potential on the y-axis, so you see negative 100 millivolts all the way to positive 50 millivolts. All right, and this dashed line is actually the threshold. So the threshold is where you got to get to this before anything's going to happen. So nothing is happening right now. The neuron is resting. So it's at resting potential. Okay, so it's pretty negative. So this is 100, that's 50. So you would say that maybe this is minus 70 or something like that. And then when these ion channels open, what's going to happen? Well, you're going to get hyperpolarized. So you can actually, you'll actually get decrease. Your inside will become more negative, and so that is shown by this dip in the graph. And that is called hyperpolarization. So you can have not just one stimulus, but two, and you can have, you know, a hyperpolarization, which is just that much. And then if you give two stimuli, then you can have an added hyperpolarization. Combined would be a bigger strength. After that's happened, you have to have equilibrium because you can't maintain a hyperpolarized state. So that's going to result in depolarization. Opening other types of ion channels triggers depolarization, which is a reduction in the magnitude of the membrane potential. Depolarization occurs if gated sodium channels open and sodium ions diffuse into the cell. So this is showing you the same idea. We just looked at the same graph, but this time now we're going to look at channels of sodium opening. And when sodium is rushing in, well, we're going to not go down because now the inside is getting more positive, not more negative. And so you're going to have a bump in the graph. If you have two stimuli, you might get a combined bump or that is higher. So that's what this graph shows you. So then that leads us to an action potential, which is actually what transmits the electrical information. The stimulus is converted into electrical information, and that is a series of hyperpolarizations and depolarizations, and that is called an action potential, and that's how the signal goes from one neuron to the next. So if the voltage inside a neuron changes by a large enough amount, an all or none electrochemical pulse called an action potential is generated. An action potential travels rapidly along the cell's axon and activates synaptic connections with other cells. And so that is the main idea of what a neuron's function is. That's what it does. And I have two beautiful links for you. Please do click on them. I'm so sorry I cannot click on them with you. The software does not allow me to do that, but do click on each of them. They're very useful for understanding an action potential and membrane potentials, and then come back to me. Now that we've looked at that, we're going to talk about what we saw and the generation of an action potential. Now this is going to be in words and me trying to explain what you saw. So first thing we know, these are facts. So neurons send messages electrochemically. We know that already. We already went over that. And we already know that the critical ions are sodium and potassium, which are one positive charge each, and calcium, which are two positive charges, and chloride, which has one negative charge, and proteins, which have negative charges, depending on how big the protein is. So we already know that because we talked about it earlier. Nerve cells are surrounded by a semi-permeable membrane. We already know that too because all cells have a semi-permeable membrane. It's a phospholipid bilayer no matter what you do. So these are facts that we already know. And then lastly, we know that when a neuron is not sending a signal, it is at rest and its inside is negative relative to the outside. So it's pretty much depolarized. So let's take a look what happens. This is what I just said in a picture. And so we look at inside the cell. And inside the cell, the things that are written in great big font are in big amounts. So there's a huge amount of proteins, which are negatively charged, and a huge amount of potassium ions. We already know that. Not so much chloride ions and not so much sodium ions. We already know that too. And then there's, of course, the semi-permeable membrane over here. And outside the cell, which is the interstitial fluid, there's an awful lot of salt or sodium chloride, but not so much potassium ions. So we already know this, and this is at rest. So I'm just going step by step. Then we're going to look at it a little bit more detail before I start generating electricity. So first, let's look at the neuron at rest. At rest, and the potassium ions can cross through the membrane easily. Yes, we know that. And chloride ions and sodium ions have a difficult time crossing the membrane at rest. We also know that because we just discussed that. And then the negatively charged protein ions, which are represented by an A with a negative superscript, inside the ions cannot cross the semi-permeable membrane, so they just keep a negative charge inside the cell. We also know that. All right. Besides these selective ion channels, there is a pump that uses energy to move 3 sodium ions out of the neuron for every 2 potassium ions it puts in. Okay, so we also looked at a pump, the sodium-potassium pump, but every time it runs, it's going to move 3 sodium ions out. Already we don't have any sodium inside the cell, but we're going to keep on moving more out. And we're going to move 2 potassium ions in in. We already have a ton of potassium in, but we're still going to do that anyway. So this is an active transport mechanism. When all these forces balance out, the difference in the voltage between the inside and the outside of the neuron is measured. And you can measure it, what is the electricity, and that is called the resting potential. So now we actually have an idea of how much charge is inside, and we can measure it. The resting potential of a neuron is about negative 70 millivolts. Oh, how nice. I actually drew that on a previous graph. This means that the inside of the neuron is 70 millivolts less than the outside. So when I put a negative over here, that's what that means. That the inside of the neuron is 70 millivolts less than the outside of the neuron. At rest, there are relatively more sodium ions outside the neuron and more potassium ions inside the neuron. We already know that, but it's nice to say it one more time. So this is how you would actually measure the voltage. You'd use a voltmeter, and you'd put electrodes inside the neuron and an electrode outside in the interstitial fluid, and you'd measure it, and you'd find the voltage difference inside and outside the neuron, which you can do. An action potential occurs when a neuron sends information down an axon away from the cell body. The action potential is an explosion of electrical activity that is created by a depolarizing current, which is a stimulus that causes the resting potential to move towards 0 millivolts. So away from negative and going towards the positive. When the depolarization reaches about negative 55 millivolts, the neuron will fire an action potential. This is ever so cool. So it goes from negative 70, and it goes up, gets to negative 55, and that's when it says, oh, well, you know what? I'm just going to make the entire action potential. I don't need to go any further. I'm just going to do the whole thing. But if it went to negative, let's say 60, or 65, nothing would happen. But when it reaches negative 55 millivolts, the neuron will fire an action potential, and that is called the threshold. So it has to reach threshold before an action potential is fired, which means the stimulus is sent. If the neuron does not reach this critical threshold level, then no action potential will fire and no information will be sent down to the next neuron. Also, whenever the threshold is reached, an action potential of the same size will always fire. All action potentials are the same size. So that is awfully cool. This leads us to the all or none principle. So therefore, the neuron either does not reach the threshold or it does. And if it does, then a full action potential is fired or it isn't. And this is called the all or none principle. So the difference in intensity of the stimulus is in how fast the signals are received. So this is an action potential, and an action potential goes like this, up and then down. So that's fine. And you would say, well, that's good. I get it. It has to get to the threshold before something will fire. But then if I feel more pain, shouldn't this be, like this? So I can say, it really hurts. No, actually, that's not how it works, because all action potentials are the same size, remember? So the only way the body can distinguish between a sharp pinprick and maybe sitting on a button is how fast those action potentials are coming. So if they're coming really, really slow, then maybe you're sitting on a button. But if you're sitting on a pin, and then the action potentials will be the same size, but they'll come really, really fast. And so that's the difference in intensity of the stimulus. How fast are the signals received, which will make the difference between how we perceive the stimulus. I do have a link at the bottom of the screen, and again, I apologize, I cannot share the link with you in real time. I can't click on it and watch it with you, which I'd love to do because it's a really good one. But please do click on it, and once you've watched it, you'll learn a little bit more of this beautiful story, and then come back to me, please. Okay, so now we're going to go through the steps of the action potential. Action potentials occur when a stimulus first causes sodium channels to open. So sodium ions are rushing on into the neuron. And we know this because there's a lot of sodium outside the neuron. Neuron, therefore, the inside of it becomes more positive with the addition of sodium ions, and so then it's depolarized. You know, it was already pretty negative, and now it's going to have all this positive stuff in, so now it's going to get depolarized. So it's going to, instead of being here, it's going to start to go like this. So when the sodium channels have opened and more sodium is coming in, the inside is not going to be as negative. And so as it gets more and more positive, we're going to go higher up. But as that is happening, the potassium channels wake up and they say, oh, we need to open too. And so it takes longer for potassium channels to open. And when they do open, then potassium is going to rush out of the cell because there's an awful lot of potassium inside the cell. And that's going to actually mess things up. So it's going to reverse the depolarization. So this is a dance. And why do the potassium channels take longer to open? Well, if you look at the periodic table, the sodium ion is a whole lot smaller than the potassium ion. So the channels are a whole lot bigger for potassium ions, and therefore they take a slightly longer time to open. So while the potassium is rushing out of the cell, unfortunately, the sodium channels start to close. Well, this makes things sort of worse, no? So this causes the action potential to go back towards negative 70 millivolts, and that's called a repolarization. The action potential actually goes past negative 70 millivolts, and that's called a hyperpolarization because the potassium channels, remember, they're too big. They open slowly and they also close slowly. So they tend to stay open for a tad bit longer. And so then you get this hyperpolarization, but then gradually the ion concentrations go back to resting levels and the cell is happy and it gets back to negative 70 millivolts. So that's what happens in an action potential. And I'll show you on this picture over here. So here we are. This is negative 70 millivolts. We already established that's the resting potential of a nerve cell. Inside the cell is more negative than the outside. All right, well, that's fine. Suddenly we get some sort of stimulus, and then the sodium channels are going to start opening. That's the first thing that happens, and that's shown in yellow. More sodium channels are opening as time progresses, and that's shown in green. Well, then the sodium channels decide that they're going to be closing, and that shows right here at the peak. The potassium channels have already opened, and now the potassium channels are going to start closing, and as they're closing, they actually take a little bit longer to close. They should just end over here, but they don't. Because they take a little bit longer to close, you get hyperpolarization, and then eventually you drift right back to negative 70 millivolts. So this is very, very cool. We're going to, again, look at these two links at the bottom of the screen. Please do click on them. They're awfully cool. So go look at them. I'm sorry I can't join you again. The software doesn't allow me to. So come back and we'll go over this in another way. So we're continuing on with the steps of the action potentials. An action potential occurs when a stimulus first causes the sodium channels to open, so sodium ions rush on into the neuron. So the neuron becomes more positive, which means it's depolarized. Now it's depolarized. It takes longer for the potassium channels to open. When they do open, potassium rushes out of the cell, reversing this depolarization. Also about this time, sodium channels start to close, and this causes the action potential to go back towards negative 70 millivolts, which is a repolarization, and the action potential actually goes past negative 70 millivolts, which is a hyperpolarization because the potassium channels stay open a little bit too long, and then gradually the ion concentrations go back to resting levels and the cell returns to negative 70 millivolts. And we looked at this once, and this is the second time we're looking at it. Okay, and this is showing you the same picture one more time. But let's go over this one more time because a lot of students have difficulty understanding the generation of action potentials. So we want to be absolutely crystal clear on it. So one more time. Graded potentials. So graded potentials are those tiny stimuli that change the polarization just a little bit. So graded potentials are changes in polarization where the magnitude of the change varies with the strength of the stimulus. These are not the nerve signals that travel along the axons, but they do have an effect on the generation of nerve signals. But they only have an effect if they reach the threshold. Only if a depolarization shifts the membrane potential sufficiently will it result in a massive change in membrane potential, which is called an action potential. Action potentials have a constant magnitude, they're all or none, and they transmit signals over long distances. So these are the three features of action potentials. They arise because some ion channels are voltage gated, opening or closing when the membrane potential passes a certain level. So action potentials only occur because of some ion channels which are voltage gated. And here is an action potential in its full glory. So here we have a negative 70 millivolts right here. This is the resting potential, and some stimulus has shown up, and you know, it's not going to fire an action potential here, it's not going to fire an action potential here, it's not going to fire an action potential here, but it will if it gets to negative 55 millivolts right here. It will fire an entire action potential. So then all those gated sodium channels are going to open. Sodium is going to rush in, and then they're going to close, and then potassium channels are going to open, and you get an action potential which is reversing. So this goes up when sodium channels are open, sodium channels close up here, and potassium channels open, and then you reverse that. The potassium channels are a little bit too clunky, so they don't close right at negative 55 or even negative 70. They actually go all the way down to maybe negative 80 or 90 millivolts. And so they do close, and ultimately you drift back to negative 70 millivolts, which is the resting potential. So here are the parts of an action potential. The first part is the resting potential. The second part is you have to get to the threshold. The third part is opening of all the sodium channels. The fourth part is opening of the potassium channels and closing of them. And then finally you get hyperpolarization, and then the resting potential back. And this is labeled in colors so you can follow along. So let's look at the numbers which correspond to these words. So generation of an action potential. An action potential can be considered as a series of stages. At resting potential, which is number 1, most voltage-gated sodium channels and potassium channels are closed. All right. When an action potential is generated, voltage-gated sodium channels open first, and sodium flows into the cell. And this would be the rising phase when in the graph the line is going up. During the rising phase, the threshold is crossed and the membrane potential increases. During the falling phase, the voltage-gated sodium channels become inactivated, and the voltage-gated potassium channels open, and potassium flows out of the cell. And here is a picture showing you those numbers again. So here is the resting phase at the bottom, and you see how the cell is negatively charged on the inside and positively charged on the outside. And then you get depolarization, and you get these sodium channels opening with sodium rushing on in. And then you get the rising phase of the action potential where a lot of sodium channels have now opened. And then you get the falling phase. So this is the same picture now. You can follow along with the numbers. So this is the falling phase, which is this one, where all the sodium channels are now actually closed. And the potassium channels are open, and now all the potassium that's inside the cell is going to be rushing out. And then you get to the undershoot, or the hyperpolarization, which is this purple part. And that is because the potassium channels couldn't get their act together fast enough, and so they didn't close fast enough, and ultimately they do close, and you get this undershoot or hyperpolarization, and it drifts back to the resting potential. So these are the same pictures shown in great big size. So you can see the inside of the cell in resting state, and then you can see the same idea in depolarization. Notice that the sodium channels are now open. And then in the rising phase of the action potential, you'll see a lot more sodium channels open. Potassium channels are still closed. And then in the falling phase of the action potential, the sodium channels are now closed, and the potassium channels are open. And then in the undershoot, the membrane permeability to potassium ions is at first higher than at rest, and then the voltage-gated potassium channels close, and the resting potential is restored. So that was the fifth part. So that's all. There is also something called a refractory period. After an action potential, a second action potential cannot be initiated, not right away. And so that tiny fraction of time is called refractory period. Refractory means contrary, he doesn't want to do it. The refractory period is a result of the temporary inactivation of the sodium channels. So because they opened and closed, now they can't open right back up, not so fast. So that's the refractory period, and at that time, no other action potential or no other stimulus will be listened to or responded to. So at the site where the action potential is generated, it's usually the axon hillock. An electrical current depolarizes the neighboring region of the axon membrane. And action potentials travel only in one direction, towards the synaptic terminals. Inactivated sodium channels behind the zone of depolarization prevent the action potential from traveling backwards, and this is why there's a refractory period. So the action potential can only go in one direction, and it can't go backwards. So the sodium channels stay deactivated for a little bit longer than necessary. And that shows you this happening over here. So here is the action potential is generated. Let's say it's over here, and you can see it's going to move down like this. So as it moves down, the sodium channels are going to be moving down, opening and closing, and the potassium is moving behind it. So there's always going to be a slight lag, and that is going to create it to be impossible for the action potential to go backwards, which is, I think, awfully cool. So now we're going to look at the speed. So how fast does it travel? Well, the speed of an action potential increases with the diameter of an axon. So the fatter the axon is, the faster the action potential is going to travel. In vertebrates, axons are insulated by a myelin sheath, which causes the speed of an action potential to increase. Whoops, I should have crossed this out. So myelin sheaths, you can consider them the rubber sheath that is around electrical wires. So if you've ever done electricity or you've looked at a wire in your house, you know that all wires are covered with some rubber, and it's either red or black. And inside that, they're copper wires. Well, why are they covered with rubber? Because rubber does not conduct. So we have myelin sheaths which are fat, and they're not going to conduct the electrical signal. Otherwise, everything around it would be zapped. So we don't want that, and we want the signal not to degrade. We want it to go in one direction. So to protect that, there is a layer of fat around the axon, so that the action potential can only travel one way and also not get degraded along the way. So myelin sheaths, they are made by glia. They're actually called oligodendrocytes, which are in this, if it's the neuron is in the central nervous system, and Schwann cells if the neuron is in the peripheral nervous system. Action potentials are formed only at the nodes of Ranvier. So this is another nice subtle distinction we're going to start looking at, because now we know what an action potential is. They're only formed at the nodes of Ranvier, which are the gaps in the myelin sheath where the voltage-gated sodium channels are actually found. All right, so that's where it's going to jump. And so they jump from node to node, and it's called saltation. Action potentials in myelinated axons jump between the nodes of Ranvier in a process called saltatory conduction, and that just means hopping, like a kangaroo. So saltation. So here we're going to look at one neuron. Here's the cell body and the dendrites, and here is a nice coating of Schwann cells or myelin sheath, and here's a little tiny area where it's uncovered, and so the action potential, which is generated at the hillock, is going to jump from here to the first node of Ranvier, then the second, then the third, and then the fourth, until it finally gets to the end of the neuron, and it's going to end up at the axon. So it's a lot faster than any chemical going, traveling all the way down like this, slowly and slowly, and dripping down, whereas electricity moves a whole lot faster. At electrical synapses, the electrical current flows from one neuron to another through gap junctions. And we looked at gap junctions before. At chemical synapses, a chemical neurotransmitter carries information between neurons. And we looked at chemical synapses before. Most synapses, of course, are chemical synapses. The presynaptic neuron synthesizes and packages the neurotransmitter in synaptic vesicles located in the synaptic terminal. The action potential causes the release of the neurotransmitter, and the neurotransmitter diffuses across the synaptic cleft and is received by the postsynaptic cell, like this. So here is the first neuron. So this is the presynaptic, and that's the postsynaptic. And we're looking right at the junction of these two. So this is the presynaptic, this is the axon, and these are the little vesicles with the neurotransmitter, and they're going to bind to the plasma membrane, drop their neurotransmitter, and the receiving neuron, which is the postsynaptic neuron, will actually get those neurotransmitters, and they will bind to its receptors, and these receptors are ligand-gated ion channels, and you can see one blown up over here. So now we're talking about the generation of postsynaptic potential. So how does the next neuron do its job? Well, direct synaptic transmission involves binding of neurotransmitters to ligand-gated ion channels in the postsynaptic cell. Neurotransmitter binding causes ion channels to open, generating a postsynaptic potential. Postsynaptic potentials fall into two categories. So you can have two responses. Either an EPSP, which is an excitatory postsynaptic potential, or an IPSP, which is an inhibitory postsynaptic potential. And just by listening to the two words, you can immediately understand why that's necessary. Because of a feedback loop, you either need to go with it or you need to shut it down. So if it's an EPSP, which is an excitatory postsynaptic potential, these are depolarizations that bring the membrane potential towards the threshold. But if it's an inhibitory postsynaptic potential, or an IPSP, these are hyperpolarizations that move the membrane potential farther away from threshold. So the signal is not going to go any further. Then we also have summation of postsynaptic potentials. So many neurons have many synapses on their dendrites and their cell bodies. And a single EPSP is usually too small to trigger an action potential in a postsynaptic neuron. So if there are two EPSPs that are produced in rapid succession, they're going to add up. And that is an effect called temporal summation. So summation just means adding up. So adding up in time. We can also have spatial summation, where EPSPs produced nearly simultaneously by different synapses on the same postsynaptic neuron can add together. So the difference is in spatial summation, the EPSPs are produced by different synapses on the same neuron, and they will add together. And the combination of the EPSPs in spatial summation and the temporal summation, these, when you add them all up, you would actually generate or trigger an action potential. You can also have negative summation, because we already talked about IPSPs, so you can add negative action potentials too. So these would be negative stimuli, and you can add them also, just like you can add a negative number. So through summation, IPSP can counter the effect of an EPSP. And so what really happens is that a receiving neuron is going to have to add up the numbers. How many EPSPs do I have? How many IPSPs do I have? And should I send the signal to the next neuron? If there are more EPSPs, yes, it will. If there are more IPSPs, no, it will not. So it adds it up, and then it subtracts the two, and the result is either generation of an action potential or not. So the summed effect of EPSPs and IPSPs determines whether an axon hillock will reach threshold and generate an action potential. And here's a picture showing you this. So it's showing you the summation of postsynaptic potentials. And so let's look at them one by one, and we can look at it. So here is E1. So this is a presynaptic neuron. Yeah. So it's touching a postsynaptic neuron. And here's another one, E2. And they're both indicated in green. But over here we have, actually, that's not an excitatory, this is an inhibitory, so that's why it's in red. So it's an IPSP, whereas these are EPSPs. E stands for EPSP, and I really means IPSP. Okay, and so what's going on? We're going to look at this graph, and so the resting potential of this neuron is negative 70 millivolts. We know that. So it gets one excitation, and it goes a little bit up, which means the sodium channels have now opened, and stuff is moving, but it doesn't get to threshold. Then you get another stimulation here, and that goes up too. So now they're both subthreshold. They're not being added up because they're just too small, and they're at different times. But if you have them at the same time, if these two EPSPs occur at the same time, they actually get added up. And so this one gets added to this, and it finally makes the threshold, and once it gets to the threshold, you get the whole action potential. In this picture over here, you can see spatial summation. So what is happening here is that the two excitatory events are actually being added up because they're occurring at the same time, and you get to the threshold and you get an action potential. But if you have the inhibition going on at the same time, and that actually minuses out some of this stuff, then you get some excitation, then you get an inhibition. And when you add them two up, you're not going to reach threshold, so nothing is going to happen. There's also modulated signaling at synapses. In some synapses, a neurotransmitter binds to a receptor that is metabotropic. In this case, movement of ions through a channel depends on one or more metabolic steps. Binding of a neurotransmitter to a metabotropic receptor activates a signal transduction pathway in the postsynaptic cell involving a second messenger. Compared to ligand-gated channels, the effects of a second messenger system has a slower onset, but it lasts longer. A single neurotransmitter may bind specifically to more than a dozen different receptors. So this is kind of interesting because usually receptors are very specific, but not for neurotransmitters. Usually they can bind to many different kinds of receptors. Receptor activation and postsynaptic response ceases when neurotransmitters are cleared from the synaptic cleft. So those are broken down rather rapidly. Neurotransmitters are removed by simple diffusion or inactivation by enzymes or recapture into the presynaptic neuron. So those things are cleared out very, very quickly. We don't want it to continue. So there's two ways of terminating neurotransmission, and it's shown in this picture. So you could have an enzyme just show up and say, okay, I got this. I'm going to break it down. So the enzyme is in green and it's going to break it down into little pieces. Or it'll actually take it back. So it's going to say, whoopsie, no, let me have you back. And so sort of like an Indian giver, it's like, I'm going to give this to you. No, I'm not going to give this to you. And so this is reuptake of the neurotransmitter by the presynaptic neuron. So you could have clearing by both methods. Oh, a very interesting neurotransmitter is called acetylcholine. It's very common in vertebrates as well as invertebrates. Very, very old molecule. It's involved in muscle stimulation. It's involved in memory formation. It's involved in learning. Vertebrates have two major classes of acetylcholine receptors, one that's ligand-gated and one that is metabotropic. A number of toxins actually disrupt acetylcholine neurotransmission, and these include the nerve gas sarin and the botulism toxin produced by certain bacteria. So botulism and sarin, these are both problems with acetylcholine disruption. Acetylcholine is just one of more than 100 known neurotransmitters. So we have a lot. We don't just have one. Acetylcholine just happens to be the biggest one. The remainder, other than acetylcholine, which is in its own a great big class, the remainder fall into four classes. So there's neurotransmitters that can be amino acids, or biogenic amines, or neuropeptides, or gases. And let's take a look at each one of these. So here is a graph that shows you, it's a table, shows you acetylcholine. Okay, so this is the biggest one, right? And this is its structure, and this is awfully cool. Or you could have amino acids. What kind? Well, you could have glutamate, or you could have GABA. You've probably heard of GABA, which is gamma-aminobutyric acid, or glycine. So these are very common amino acids which will actually act as neurotransmitters. So here we are looking at amino acids, which we looked at much earlier on in Bio 101. We said they have all these properties. Yes, they do. And then we have amines, and we looked at these in the immune system. I beg your pardon, in the hormonal, the endocrine system, and we saw that the amines were epinephrine, norepinephrine. Well, this is what they look like. And then there's also another amine called dopamine and another one called serotonin. And notice how similar these structures are. So similar. Difference of just one OH that's missing over here. And then this serotonin has actually a 5-ring compound here. Which is a little bit different than the others. So very, very similar molecules, but very potent. And then there's neuropeptides, which is a very diverse group, and there are only two that are shown over here. There's substance P, and actually what they're showing you is the sequence of the amino acids. And then there's methamphetamine, which is an endorphin, and it's just 5 amino acids long. And then there's gases like nitric oxide. Nitric oxide is actually a really cool gas, and it's used in many, many functions in our body, including being a neurotransmitter, which you wouldn't think. So let's look at the amino acids first. Amino acid neurotransmitters are active in the central nervous system as well as the peripheral nervous system, and they're known to function in the central nervous system as glutamate and GABA and glycine. The biogenic amines include epinephrine, norepinephrine, dopamine, serotonin, and they are active in both the central nervous system and the peripheral nervous system. The neuropeptides, several of them, the relatively short chains of amino acids, also function as neurotransmitters. Neuropeptides include substance P and endorphins, which both affect our perception of pain. Opiates can bind to the same receptors as endorphins and can be used as painkillers. And the receptors, remember, are not so very specific. So that's why opiates, which are similar shaped like endorphins, are mistaken by the body and can bind to those same receptors that were meant for endorphins. And the gases like nitric oxide, which is a very good one, and carbon monoxide, that's also used, believe it or not, even though it's poisonous. Both these gases are poisonous. And they're local regulators in the peripheral nervous system. Unlike most neurotransmitters, nitric oxide is not stored in cytoplasmic vesicles, but is synthesized on demand. It's that bad. You can't keep it, so you have to make it when you need it. It is broken down within a few seconds of production, and although inhaling carbon monoxide can be deadly, the vertebrate body synthesizes small amounts of it, some of which is used as a neurotransmitter. So small amounts are okay of carbon monoxide, but not for nitric oxide. All right, so now we can go and look at the types of neurons. Neurons connect to each other and form neural networks. Specialized types of neurons include sensory neurons that respond to stimuli from sensory organs, or motor neurons that respond to signals from the brain. Whoops, this should be from the brain and make muscles contract. And interneurons that connect to each other in neural networks. So there are three types of neurons, and they're depicted over here pictorially. So here is a sensory neuron, here is a cell body, and look how long its axon. Actually, it has two, and that would be a sensory neuron. And then there are interneurons, and you can see that the cell body is here, but there are an awful lot of dendrites, and so that would be a characteristic of an interneuron. And then you see a motor neuron. It has one very long foot and a lot of dendrites. The human brain contains about 100 billion neurons, and they're organized into circuits more complex than the most powerful supercomputers. Each single-celled organism can respond to stimuli in its environment. Animals are multicellular and respond to stimuli using systems of neurons. So let's look at the command and control center. The simplest animals with nervous systems, which would be the cnidarians, have neurons arranged in nerve nets, which lacks cephalization. So cephalization means head, so there's no like head of the animal. So they just have nerve nets, so no brain in that sense. These nerve nets emerged before the Cambrian explosion about 570 million years ago. These are very old. A nerve net is a series of interconnected nerve cells. So sea stars, which are really pretty, have a nerve net in each arm connected by radial nerves to a central nerve ring. So now these are getting a little bit more complex because now you have a central nerve ring, which is kind of sort of like a primitive brain. More complex animals have nerves, not a net, and nerves are bundles that consist of the axons of multiple nerve cells. And let's look at them. So here is a nerve net in a cnidarian. So this would be Hydra. So it doesn't have a brain, but it does have many neurons that are covering most of the body. So if you happen to poke it like dead here, it won't feel it. Then we have the sea star, which has these radial nerves, which are all connected to a nerve ring, which is central. So that already gives you a simple central nervous system. And that starts in bilaterally symmetrical animals, which exhibit cephalization, which is the clustering of sensory organs at the front end of the body. Relatively simple cephalized animals, such as flatworms, have a central nervous system, believe it or not, even though that we think they're nothing, but they actually are pretty smart. The central nervous system consists of a brain and longitudinal nerve cords. Annelids and arthropods, so these are bugs, have segmented arranged clusters of neurons called ganglia. And here is a picture showing you a planarian. Notice here he has a very, very primitive brain. He does have nerve cords, and then there are nerves coming off of it. So 2 nerve cords. And then we have an annelid like a leech. He too has a very, 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 very small brain, but now he doesn't have 2 nerve cords, and he actually just has the 1. And we're seeing increasing complexity, and then of course there are nerves coming off of it. And then we go up to arthropods, and there's actually a definite small brain, and there is now just the 1 nerve cord, which is central, and it will segment in every segment of the animal. And then we have, if you look at this next animal, which is a mollusk, it has an anterior nerve ring, which is kind of sort of like a brain, and then it has a whole bunch of longitudinal nerve cords, so we're going backwards in this one in complexity. Nervous system organization usually correlates with lifestyle. Sessile mollusks, like clams and chitons, which we just looked at, have very simple systems. They don't need a complex system, so why bother building one? Whereas more complex mollusks, like octopus and squid, they have very sophisticated systems. So here we have a squid, and he actually has a definite brain, and he has ganglia, and he has nerves going down every arm of him. And then we go to vertebrates, and you actually see that there's this cephalization part is pretty prominent. The head, the brain is located in the head, and it's pretty big compared to what we've seen before. And then we have one spinal cord, which is moving all the way down the animal, and then nerves branch off every part as in segments, although he doesn't have any segments. So let's look at the organization of the vertebrate nervous system. In vertebrates, the central nervous system is composed of the brain and the spinal cord, and the peripheral nervous system, or the PNS, is composed of nerves and ganglia. The brain and the spinal cord contain gray matter, which consists of neuron cell bodies, dendrites, and unmyelinated axons. White matter, which consists of bundles of myelinated axons, so this is all the fat. And here's a picture of it. So if that's our brain, and we cut it this away, and then we looked into it, well, we see the gray matter, which are all the very important cells sticking on the outside. And then all the fat is in the inside, which is the white matter. Oh, yes, and these are the 3 ventricles. We have 3 holes in our head called ventricles. Here's the central nervous system, and you see a really, really enlarged brain. You see a spinal cord, which ends kind of over here. And then you see the PNS, which are all these nerves that are outside the CNS. So the spinal cord can actually think on its own, and it doesn't necessarily have to send all its information to the brain. Spinal cord conveys information from and to the brain, but the spinal cord also produces reflexes independently of the brain. A reflex is the body's automatic response to a stimulus. For example, when a doctor uses a wooden hammer to hit you on your knee, you get a knee-jerk reflex, and that is never felt by you as thinking, because the message never gets to the brain, and the knee-jerk reaction is something that is just conveyed from the spinal cord. This is the picture of the spinal cord just taking care of the business instead of telling the brain to do something. The cerebrospinal fluid, this is another very important part of the nervous system. The central canal of the spinal cord and the ventricles of the brain are hollow, and they're filled with a fluid called the cerebrospinal fluid. The cerebrospinal fluid is filtered from the blood, and it functions to cushion the brain and the spinal cord and to provide nutrients and also to remove waste. So basically, it's the interstitial fluid of the central nervous system. Glia, so they have numerous functions, and those include nourishing and supporting and regulating neurons. Embryonic radial glia form tracks along which newly formed neurons will migrate. So they start their functions early on in embryology, when you still are, you have the embryo forming, the cells are still moving around, and you have a blastula and a gastrula. Well, at that point, you're going to have these little glia cells which are going to form tracks once the nerve cells have differentiated, and tell the nerves to follow us to this particular area where we're going to make the brain. Astrocytes are also support cells, and they induce cells lining capillaries in the central nervous system to form tight junctions, resulting in a blood-brain barrier, which is never breached, and restricting the entry of most substances into the brain. So we need a blood-brain barrier because we may eat a whole bunch of stuff, and we don't need all of that mess to go to the brain. We need only the good stuff. So most bacteria, everything is cut off. And so that is cut off because they're very tight junctions that are in these capillaries that actually supply blood to the brain. And those are controlled by astrocytes. They make those so that we don't get sick. We never get bacteria in the brain. That's one place that bacteria cannot go. Here is a picture showing you this. So you see the central nervous system, which goes up to here, and then the peripheral nervous system. So these would be all the nerves. And in the central nervous system, you'll see the astrocytes telling the blood vessels, okay, you know what, let's make a blood-brain barrier. And then you'll see all sorts of the neuron cell bodies and the glia, and everybody helping the nerve cells, the neurons, to function. The peripheral nervous system. The PNS transmits information to and from the CNS, and it regulates movement and the internal environment. In the PNS, afferent neurons transmit information to the central nervous system, and efferent neurons transmit information away from the central nervous system. So please remember the spelling of these two, because you miss one and you'll have the complete wrong definition. There are actually two efferent parts of the peripheral nervous system, the motor system and the autonomic nervous system. The motor system carries signals to the skeletal muscles, and it's voluntary. And then there's an autonomic nervous system, which is part of the PNS. It regulates the smooth and cardiac muscles, and it's generally involuntary, and you have no control over that. All right, so let's look at this graph. It shows you the central nervous system, and its job is to process information. Got that? Well, where does it get the information from? Well, it gets it from internal and external stimuli, which are sent by sensory receptors, taken up by afferent neurons, and afferent neurons go to the central nervous system. The central nervous system processes all that information and sorts it out, and then sends a response by its efferent neurons, either to the autonomic nervous system or the motor system. If it's the motor system, then that's the control of skeletal muscle. If it's the autonomic nervous system, well, the autonomic nervous system actually has three divisions. There's a sympathetic division, a parasympathetic division, and an enteric division, and they're all involuntary. We do not control them. They control all sorts of muscles that are inside us that we never ever control. So let's look at the autonomic nervous system. Remember, it has three divisions. It has the sympathetic, parasympathetic, and enteric divisions. Well, let's look at what does each division do. The sympathetic division of the autonomic nervous system regulates arousal and energy generation and the fight or flight response. The parasympathetic division of the autonomic nervous system has antagonistic effects on target organs and produces calming and a return to rest and digestive functions. So it does the opposite of the sympathetic. So we looked at the sympathetic nervous system. We touched on it in the endocrine system. We looked at the flight or fight response, and we said that the sympathetic nervous system actually controls part of that, and here we are, we're actually talking about it, which is very nice. And then the third part is the enteric division, and that controls the digestive system. So it will control the digestive tract, the pancreas, and the gallbladder. And here are here's a nice diagram which divides up the parasympathetic system of our nervous system and the sympathetic division, and it tells you what each does. So if you look at the parasympathetic division, it acts on target organs like your eyes. It will constrict it. It will stimulate salivary gland secretion. It will constrict bronchi and lungs. It will slow down your heart. It will stimulate activity of the stomach and the intestines, and stimulate the activity of the pancreas, and stimulate activity of the gallbladder. So it's the rest and digest function, whereas the sympathetic division actually will dilate the pupil of the eye so you can see more, see better. You don't need any salivary gland secretions because you're about to do something, you're running or fighting. It will relax the bronchi and the lungs so you can take in more air. It will speed up the heart, and it will inhibit the activity of the stomach and intestines because we're not really concerned about digesting our food right now. We want to run away or we want to fight. So it's going to do all sorts of activities that promote the fighting response. So this would be the rest, and this is the fight. All right. And here is the same picture, but written in a smaller, a bigger font, so you can look at it and you can see what is going on and where it's going on. Sometimes you have ganglia, and sometimes you actually have nerves. So here's the second part of it. Notice that the sympathetic division generally is in ganglia. All right, and then we have the regionally specialized vertebrate brain. So this is awfully cool because now we're actually going to look at the brain. Specific brain structures are particularly specialized for diverse functions. These structures arise during embryonic development. Invertebrates usually have a ventral nerve cord, while vertebrates usually have a dorsal spinal cord. So that's one difference. The spinal cord and brain develop from the embryonic nerve cord. The nerve cord gives rise to the central canal and the ventricles of the brain. So here is, when we looked in embryology, we left off at a squiggle of an embryo, and we said, well, here is the ectoderm, and it's going to make its brain. And that's true. So part of it, the front part, will actually differentiate into the forebrain. Then you have the purple part, which is going to be the midbrain, and then the blue part, which is the hindbrain. And as the embryo develops bigger and bigger, you see where those cells end up. And you can look at the fully formed baby over here. You can see that actually before that, what's all that yellow stuff? That was never part of this story. Well, there's this extension of the forebrain which is yellow, so there's an extra part added on, and what you see is that small extension actually becomes a cerebrum, or the entire brain of the human, and the forebrain shrinks down into this very small area which has the hypothalamus and the part of the pituitary. And then the purple part, which is the midbrain, also becomes very small and shrinks down. And then the blue part, which was this giant hindbrain, actually becomes the medulla oblongata, which controls our breathing, and the spinal cord. And we generate something extra, which is the cerebellum for balance. So this is kind of cool. And here are pictures of the same picture, but blown up so you can look at it in more detail. And this is also blown up, and so you can look at it in more detail. And here is a picture. This is the adult brain looked at from the rear. And so now you can see that there is actually a right and left hemisphere of the brain. And then there are these fibers that connect the right and left brain together. And those are called the corpus callosum. And they result in the two sides of the brain talking to each other. And this is the cerebellum, which is responsible for our balance. And here is another picture of the brain, a side view, and it shows you the forebrain, which in embryonic terms was just the yellow part, and then the midbrain, which is shrunk down into the center, and the hindbrain, which is the blue part and becomes all these other areas that are left over. Here you see the brain itself. So we're going to look at the cerebrum and look at all the folds of it and see, well, which areas do what. And when you look at the folds, and it looks like a big mess, but it's actually not. And if you look at the front part of it, that is the frontal lobe. And then right behind it is the parietal lobe. And then underneath these two are the temporal lobe. And then right behind it, at the very end of our head, is the occipital lobe. So really not a big deal. It's just knowing the four lobes, which are frontal, parietal, temporal, and occipital. And here are other pictures which will tell you what do they do, because you always want to know what part of the brain does what. And you can see that the occipital lobe is actually responsible for sight. So that's very important. It's for sight, for image recognition. And then when you see this part, the temporal lobe, you can understand that it's responsible for short-term memory, for emotions, and for equilibrium. This yellow strip, that is the motor function. So that controls our voluntary muscles. And this blue part right behind it is actually the sensory part that controls the sensation from skin and muscles. And then you look at this area called Broca's area. This is where how we talk. And then if you look at this area, which is right in the front, that is where all our higher thinking goes on. We think, we concentrate, we plan, we judge, we create. And that's where all the executive functions happen. And then if we look at the purple area, which is here, and this is eye movement and orientation. So notice that we have a huge area responsible for eyes. We're devoting a huge amount of brain real estate to just eyes, so we can take in information properly. And then this tiny blue area over here, this is the somatosensory association area. So we can evaluate here. This is where we think what is the weight, what is the texture, what is the temperature, and so on, and so we can recognize objects. So that's the part that recognizes objects. And then if we look at Wernicke's area, which is this pinkish area right here, that is where we can read and speak a language. So that's where we understand the stuff that we're talking about. And of course, the cerebellum is for balance and coordination of movement, and that's about it. And if you actually cut this brain, or you can see these areas reflected that we just talked about, or if you have a view, if you look down the brain and it's not cut, the same areas are shown in a superior view. If you look underneath the brain, which is an inferior view, you would see these areas. So it's kind of cool to look at it from every which way. So let's look at arousal and sleep. The brainstem and cerebrum control arousal and sleep. The core of the brainstem has a diffuse network of neurons called the reticular formation, and this regulates the amount and the type of information that reaches the cerebral cortex and affects alertness. And the hormone melatonin, which we looked at previously, is released by the pineal gland and plays a role in bird and mammal sleep cycles. So this is awfully cool. Now it's all coming together. Here is a view of the brain which shows you what is going on. So we're taking information from the eye, yes. The optic nerve is going to take information which is going to radiate through all parts of the brain. We're also going to take information from our ears, yes. And we're also going to take information from touch, pain, temperature, and so on. And it all gets integrated inside our brain, and we look in the reticular formation where this is all integrated and then ultimately radiated out so appropriate action can happen. Sleep is essential and is thought to play a role in the consolidation of learning and memory. Dolphins sleep with one brain hemisphere at a time and therefore are able to swim while they're asleep, which I think is fascinating. And this shows you the dolphin's brain when it's sleeping. So when the left hemisphere is actually sleeping and you get these pretty waves, the right hemisphere is very, very active. And then when the left hemisphere is very, very active, the right hemisphere is sleeping. So these show you how the dolphin brain works. We also have cycles, and these are circadian rhythms. We talked about circadian rhythms earlier, and sleep and wakefulness are cycles of circadian rhythms. These are daily cycles of biological activity. Mammalian circadian rhythms rely on a biological clock, which is a molecular mechanism that directs periodic gene expression. Biological clocks are typically synchronized to light and dark cycles. In mammals, circadian rhythms are coordinated by a group of neurons in the hypothalamus called the SCN, or the suprachiasmatic nucleus. The SCN acts as a pacemaker, just like we have a pacemaker on the heart. The SA node, we have an SCN nucleus that sits on top of the hypothalamus, and it synchronizes the biological clock, which I think is awfully cool. And then the limbic system. The limbic system is responsible for emotions. So the generation and experience of emotions involve many brain structures, including the amygdala, the hippocampus, and parts of the thalamus. And these structures are grouped as the limbic system. So the limbic system is the amygdala, the hippocampus, and parts of the thalamus. So these are all responsible for emotion. And the limbic system also functions in motivation, in olfaction, in behavior, and in memory. And here it is. This is the limbic system highlighted in orange. So you have the amygdala over here, the hippocampus here, the hypothalamus we already know, and then the thalamus. So these are all parts of the limbic system, and yes, the olfactory bulb feeds into it. So this is very important for it. The generation experience of emotions requires interaction between the limbic system and the sensory areas of the cerebrum. Storage of emotion in the memory is done in the amygdala, which is a massive nuclei near the base of the cerebrum. And we know this because if we play happy music, you see the nucleus accumbens here light up, and you see other areas light up. But when we play sad music, the amygdala lights up. So this is very interesting. So we have proof on this. All right, so here is this cerebrum. We're going to go for it one more time. This time it's a great big picture with some words. So we have the frontal lobe. This is where all the decisions are made and executive stuff is going on. The first stripe of it is the motor cortex, and then right behind it is another stripe of the parietal lobe, and that's the somatosensory cortex. And the parietal lobe is responsible for sensory association. It integrates all kinds of senses. Inside the frontal lobe is Broca's area, which is how we talk. And then we have the temporal lobe. And remember that we have Wernicke's area, which is here. And then we have auditory cortex, which is here, which is how we hear. And then the temporal lobe is underneath the frontal and the parietal lobe. And then we have the pink area in the back, and that pink area is the occipital lobe, which is completely responsible for visual association and our vision. It's the visual cortex, and it recognizes patterns and stimuli that are visual. And underneath, tucked it all, is underneath the cerebrum is the cerebellum, and this again is the spinal cord. Language and speech. Studies of brain activity have mapped areas responsible for language and speech. Broca's area in the frontal lobe is active when speech is generated. Wernicke's area in the temporal lobe is active when speech is heard. These areas belong to a larger network of regions involved in language. And so this is our brain lighting up. When you hear words, you see different areas light up. When you see words, which is reading, and so notice that this is the back of the brain. And then when you're speaking words and you're hearing them, when you're actually speaking them and not just hearing them, this area lights up. But when you're talking, when you're actually talking, that's the area that generates words. So very, very cool. Very important to know is that there are two hemispheres of the cerebrum. There's a right and left hemisphere. Well, the two hemispheres make distinct contributions to brain function. The left hemisphere is more adept at language, math, logic, and processing of serial sequences. But the right hemisphere is stronger at pattern recognition, nonverbal thinking, and emotional processing. And this is called lateralization of cortical function. This differences in hemisphere function is called lateralization. Lateralization is partly linked to handedness. The two hemispheres work together by communicating through the fibers of the corpus callosum, which I showed you in a previous picture. And here it is again. So here is the right hemisphere, the left hemisphere, and then you have these tissues that connect the two, and that is the corpus callosum. The corpus callosum allows for communication between both hemispheres. The corpus callosum transfers motor, sensory, and cognitive information between the two hemispheres. The cerebral cortex receives input from sensory organs and somatosensory receptors. And this is how it's going to process the information. The somatosensory receptors provide information about touch, pain, pressure, temperature, and the position of muscles and limbs, so where you are in space. The thalamus directs different types of input to distinct locations. The thalamus has an awfully big role in all this. Information processing, we're continuing on with this. So adjacent areas process features in the sensory input and integrate information from different sensory areas. Integrated sensory information passes through the prefrontal cortex, which helps plan actions and movements. In the somatosensory cortex and motor cortex, neurons are arranged according to the part of the body that generates input or receives commands. Frontal lobe function. So let's check it out. If there's damage to the frontal lobe, because it does executive decisions, that's going to impair decision making and emotional response. But it might leave the intellect and the memory intact. It really depends on where the damage is. The frontal lobes have a substantial effect on executive functions. Oh, memory and learning. So this is a good one because we're always interested in memory and learning. Two processes dominate embryonic development of the nervous system. The neurons, and there's a lot of them, they compete for growth supporting factors in order to survive. So there's, just that much food and space, and there's a lot of neurons. So that's how the embryo gets rid of a lot of the neurons. There's not enough food. Only half the synapses that form during the embryo development survive into adulthood. So children that are about 3 years old have about 15,000 synapses per neuron. That's a lot. But adults only have about half that number, about 7,000 synapses per neuron. So that's still a lot. But not just the neurons are trimmed off, but even the synapses are trimmed off as we grow older. Neural plasticity is the ability of the nervous system to be modified after birth. And this is important because that shapes our brain and our learning abilities. Changes can strengthen or weaken signaling at a synapse, so we can learn. Here is a picture showing you synapses being strengthened or weakened in response to activity. So it shows you in picture A, there is a neuron and there is some sort of signal that is going on, and if you keep on doing that signal, you might actually develop a bigger area that's going to be devoted to that signal, and so these branches will grow into bigger branches, and so you're strengthening that activity. If they're both active at the same time, then if there are two synapses often active at the same time, the strength of the postsynaptic response may increase at both synapses. So this is how we learn at the cellular level. The formation of memories is an example of neural plasticity. Short-term memory is accessed via the hippocampus. So do remember that. The hippocampus also plays a role in forming long-term memory. Long-term memory is stored in the cerebral cortex. Some consolidation of memory is thought to occur during sleep. So some of our short-term memories are shunted off to the cerebral cortex to be stored forever. And that's done during sleep. Long-term potentiation, one of my favorites. In the vertebrate brain, a form of learning called long-term potentiation involves an increase in the strength of the synaptic transmission. Long-term potentiation involves glutamate receptors, and we looked at glutamate a little bit earlier. If the presynaptic and postsynaptic neurons are stimulated at the same time, the set of receptors present on the postsynaptic membranes changes. So this is so cool. Memories are encoded by the strength of the synapses, so long-term potentiation is one of the main mechanisms underlying memory and learning. And so when I do say go over it again and again, I'm actually telling you something that is supported by science. And this does happen. So we can actually encode our memories by doing it over and over and over and over again. The number of neurons, which we have, which is 100 billion, does not change. So memories or learning are not the result of new neuron production. Instead, they're formed by strengthening the connections between existing neurons to improve their effectiveness. And then neurons that fire together are usually wired together, and that's how we get a memory back. So we keep on strengthening those synapses, we will remember things much faster. Here is a cartoon version of showing you how we have long-term potentiation. So we have a presynaptic neuron right here, and it is giving out glutamate, and we have these receptors which are going to actually in the postsynaptic neuron take them up. Because they are going to take them up, we have then a loop being established. We're going to store this glutamate in an AMPA receptor, and the AMPA receptor is going to sit up top waiting for that signal to come back again. And if you get the signal again, like you read the book again, you'll get maybe another AMPA receptor sitting there. And so this is called long-term potentiation, or the building of memories, and the foundation of learning. So here is the same idea shown in step by step. So you can actually see these AMPA receptors and learning happening and memories being formed. Here is a sketch showing you what happens in these areas that I mentioned before. So this is the parietal lobe, and this is the somatosensory cortex. And this is that strip, the second strip, not the first strip. The first strip was the motor sensory cortex. And what is happening over here is that if you prick your finger with a pin, the pain receptor in the finger will send that message by an afferent sensory neuron to the spinal cord, and there will be interneurons that are connected, and you will immediately pull your hand back. And that will happen rather fast because the efferent motor neurons will immediately take it. But another path of it is actually going to go to the parietal lobe, and it will be perceived as pain. So that is how something as simple as pricking your pin works. Here are two links that I have picked for you, which I thought would be kind of cool to look at. The second link is just for fun, but please do look at them. I'm sorry again, I cannot join you in looking at these links. I'd love to, but the software I have does not allow me to. So check them out and come back to me. Transduction and transmission to the central nervous system. Let's talk about this. So all stimuli represent forms of energy. Sensation involves converting energy into a change in the membrane potential of sensory receptors. When a stimulus's input to the nervous system is processed, a motor response can be generated, like a simple reflex or more elaborate processing. So, for instance, here is a mole, and he's looking for food. If he doesn't see food and he's in a tunnel, he's just going to keep on moving on. So he gets a sensory input, no food, integrates that message, and he says, okay, fine. And the motor response is keep moving. But if he does see food, so that's the sensory input, he understands that. He integrates that, and he says, must eat it. So then instead of moving on, he actually bites it. So now we're going to look at sensory pathways. Sensory pathways have four basic functions in common. There's, first of all, and these should be, this is a pattern, I keep repeating myself. These are the same four basic functions I keep on talking about again and again. First, you have to receive the stimulus, then you have to transduce it, then you must transmit it, and then you have to integrate it. Sensory reception and transduction. How do we do it? So we have sensory receptors which interact directly with the stimuli, which could be internal stimuli or external stimuli. So we have these receptors. Then we have sensory transduction, which is the conversion of the stimulus energy into a change in the membrane potential of a sensory receptor. That's how we understand the world around us. This change in the membrane potential is called a receptor potential. Receptor potentials are graded potentials. So it won't necessarily result in an all or none response, but they're graded. So their magnitude varies with the strength of the stimulus. After the energy has been transduced into a receptor potential, some sensory cells generate the transmission of action potentials to the central nervous system. Some sensory receptors are specialized neurons, while others are specialized cells that regulate neurons. Sensory neurons produce action potentials, and their axons extend into the central nervous system. All right, so here is a generic sensory receptor, and the receptor is afferent. And so a stimulus happens, could be internal, external, and the stimulus is, the message is sent to the central nervous system. Okay, so that would be an afferent neuron. Here on this second panel, what you see is a stimulus, happening to a sensory receptor cell. And this is an actual cell, and it's going to actually generate some neurotransmitter, and the neurotransmitter is going to be released, and it is picked up by an afferent neuron, which will then send the message to the central nervous system. So here a receptor is regulating the afferent neuron, whereas the first panel here, the receptor is indeed the afferent neuron itself. So there is a choice. Should there be an action potential, or should we use a neurotransmitter? So the response of a sensory receptor varies with the intensity of the stimuli. If the receptor is a neuron, a larger receptor potential results in more frequent action potentials. But if the receptor is not a neuron, a larger receptor potential causes more neurotransmitters to be released. And that's why we have sensory organs. Perceptions are the brain's construction of stimuli. So what we perceive as a stimulus does not necessarily mean that the other person perceives it in the same way, or the other animal perceives it in the same way. Stimuli are different sensory receptors. Stimuli from different sensory receptors travel as action potentials along dedicated neural pathways, and the brain distinguishes stimuli from different receptors based on the area in the brain where these action potentials show up, wherever they show up, and that's how we can tell what are we looking at. Is it something visual or auditory, or is it pain, or what? Amplification is the strengthening of the stimulus energy by cells in sensory pathways. So we always amplify the signal. And then sensory adaptation is a decrease in responsiveness to continued stimulation. And what does that mean? So if we sit on like a stone, initially it'll be kind of pokey and you'll feel uncomfortable. But then a sensory adaptation occurs, and you don't get that stimulus anymore. The brain figures out that we're not going to be moving, so might as well cut off the pain cells, pain response, because it's not that bad. So let's look at mechanoreceptors first, because they were first on the list. Mechanoreceptors are very cool. They sense physical deformation caused by stimuli such as pressure, or stretch, or motion, or sound. So that's a whole bunch of stuff. The knee-jerk response is triggered by the vertebrate stretch receptor, which is a mechanoreceptor that detects muscle movement. And we have these very special cells called Pacinian corpuscles. These are nerve endings in the skin responsible for sensitivity to vibration and to pressure. So that's how we feel texture. These corpuscles are especially sensitive to vibrations, which they can sense even centimeters away. And believe it or not, some of them are in our pancreas. So I'm not sure why they're there, but they are. Pacinial corpuscles, again, the vibrational role of these corpuscles is to detect the surface texture. So that's how we can feel if something is satiny smooth or coarse, like cardboard, rough versus smooth. The Pacinian corpuscles are also found in the pancreas, which I just said, where they detect vibration and possibly very low frequency sounds, and possibly contributing to our tummy aches. Pacinian corpuscles are about 1 millimeter in length, so they're pretty big. 1 millimeter you can actually see with the naked eye. They're oval shaped, and they have about 20 to 60 concentric lamellae of fibrous connective tissue and fibroblasts separated by gelatinous material, all wrapped in a layer of connective tissue. Let me show you a picture. Like here. The lamellae are very thin, flat, modified Schwann cells. So if you follow one over here, this just keeps on going up, and then you go around, and you go around, and you go around, and you keep on going around, and you finally get to, this area. The center of the corpuscle is the inner bulb, which is a fluid-filled cavity with a single afferent unmyelinated nerve ending. Deformation in the corpuscle causes pressure-sensitive sodium channels in the axon to open, allowing sodium ions to influx, generating an action potential. So if we squash it, then this is all going to get squashed. and this will push this, and then you get an action potential. That's how you can tell. Hearing and perception of body equilibrium are related in most animals. For both senses, settling particles or moving fluid is detected by mechanoreceptors. Most invertebrates maintain equilibrium using mechanoreceptors located in organs called statocysts. Statocysts contain mechanoreceptors that detect the movement of granules called statoliths. Let's look at them. So here we have a statocyst with statoliths. So these are the rocks that are inside of tiny rocks that are sitting inside a little ball of cells. And you can see these are receptor cells with little hair that are sticking out. And because these little balls of lithified stuff is like little stones sits on them, and when they move, they give information which is taken by the sensory nerve fibers. Many arthropods sense sounds with body hairs that vibrate with localized ears consisting of a tympanic membrane and receptor cells, and they're usually on their legs, so they hear with their legs. Hearing, very, very interesting. So this is a mechanoreceptor sense. Vibrating objects create percussion waves in the air, which cause the tympanic membrane to vibrate. The three bones of the middle ear transmit the vibrations of moving air to the oval window on the cochlea, and these vibrations create pressure waves in the fluid in the cochlea that travel through the vestibular canal. And you've seen this in lower grades, but let's look at it one more time. The fluid waves dissipate when they strike the round window at the end of the tympanic canal, and pressure waves in the canal cause the basilar membrane to vibrate, bending its hair cells, and this bending of hair cells depolarizes the membranes of the mechanoreceptors and sends action potentials to the brain via the auditory nerve. As so. So here we have the ear, and here are pressure waves going all the way to the tympanic membrane, which is attached to three tiny bones. And these are the smallest bones in your body. This is attached to the round window, which in turn is attached to the cochlea. And the cochlea is a series of curled cells, curled tubes, and they are attached to three semicircular canals as well, which give you balance. And inside the cochlea, if you actually opened it up, you would see the auditory nerve here, and in the very dead center of it is an organ of Corti, which has little hair. And let's look at it. And so here's a picture, and then here's another picture. So this picture actually shows you the organ of Corti, and it shows you the nerve cells, the actual hair cells in yellow with the hair, which are touching a tectorial membrane. The basilar membrane is this part at the bottom which lines it, and that these hair are blown up. So this part is blown up to show you these are the tiny things that move with the percussion waves that allow us to hear in our brain. This is all blown up to show you. The ear conveys information about volume, which is the amplitude of the sound wave. Amplitude is how high. And pitch, which is the frequency of the sound wave. So how frequent are these? The cochlea can distinguish pitch because the basilar membrane is not uniform along its length. And we saw that in the previous slide. We saw sometimes it's bunched up in like 2 or 3 layers of cells, and sometimes it's a single layer. Each region of the basilar membrane is tuned to a particular vibration frequency, and this is the cool part. So the basilar membrane can distinguish pitch, and so here is the basilar membrane, yeah, and in different areas it's going to be different pitches. So we can see it here a little bit better, and here a little bit better. And here is the actual work of it. So here is a sensory neuron. So first of all, we have this, which is the basilar membrane cell, and here are the hairs. If they're not moving, then neurotransmitter is being sent at the synapse, and the signal is just not going to go. You know, it's just flat. But if there is a signal, then it would actually produce an action potential. If you have the bending of the hairs in this direction, you can sense it, because the signal is then sent in this way that there is a depolarization, and it is shown as these action potentials. But if the hair are bent that way, then less neurotransmitter is produced, so that produces hyperpolarization, and that is how we actually sense it. So one way will produce depolarization, the other side will produce hyperpolarization, and we can tell where we are in space. Equilibrium. So several organs of the inner ear detect body movement, position, and balance. The utricle and the saccule contain granules called otoliths that allow us to perceive position relative to gravity or to linear movement. 3 semicircular canals contain fluid and can detect angular movement in any direction. So these are our 3 semicircular canals. Yes, 1, 2, 3, and they're in 3 different directions. So this is the XY direction, we all know, and then the Z direction. So we always know where we are in space. What is inside these are actually, so here is some epithelial cells, and they have little hairs on them, and on top of them is a little cupula, so there is lymph flowing inside, and it will tell us which way are we standing or headed. Unlike mammals, fishes have only a pair of inner ears near the brain. They don't have external ears. Most fishes and aquatic amphibians also have a lateral line system along both sides of their body. That would be the line that you see in the center of the fish. So if this is a fish, so this line that you actually always take out before eating it. And this lateral line system contains mechanoreceptors with hair cells that detect and respond to water movement. Here we are. I didn't have to draw it. So that's the lateral line showing you, and it has the same idea. So although we thought that we're all that with our semicircular canals and a cupula, it's the same pattern that we see in a fish, a lowly fish. Same pattern. Chemoreceptors. They transmit information about the total solute concentration of a solution. Now this is really important because we want to keep our internal chemical environment kind of constant. When a stimulus molecule binds to a chemoreceptor, the chemoreceptor becomes more or less permeable to that ion. Then there are electromagnetic receptors. They detect electromagnetic energy such as light, electricity, and magnetism. Some snakes have very sensitive infrared receptors that detect body heat of prey against a colder background, and that's because snakes are on the ground, so they can't stand up and look around. And other animals use the Earth's magnetic field to migrate and to orient themselves, and so you might wonder, how do the birds always know how to come back home? There are no maps, they don't no GPS, nothing. They use their electromagnetic receptors, and they orient themselves to the Earth's magnetic field, which has invisible lines, and they manage to find their way every single time. And they could be monarch butterflies, or they could be birds, but they can find their way much better than we can, or I can. Here is a rattlesnake, and that is his infrared receptor. It's not his nose, so that's how he senses prey. Thermoreceptors, which respond to heat or to cold, help regulate body temperature by signaling both surface and body core temperature. So we have a core temperature, and then we have the rest of the body temperature. And we have thermoreceptors which help us maintain our core temperature. If that goes below a certain limit, then there's not much that can be done to save that organism. Mammals have a number of kinds of thermoreceptors, each specific for a particular temperature range. And this is because our enzymes have to work in certain temperatures. Pain receptors. So pain receptors are also called nociceptors, and in humans they are a class of naked dendrites in the epidermis. So they're just the dendrite of a neuron sticking out. They respond to excess heat or pressure or chemicals released from damaged or inflamed tissues. Visual receptors, now these are good ones. Animals use a diverse set of organs for vision, but the underlying mechanism for capturing light is the same, pointing to a common evolutionary origin. Just like we saw the fish with its hair and cupula, and we have the same for our equilibrium. We see that same pattern. So nature evolved it once, and then there are just modifications in all animals. Light detectors range from simple clusters of cells that detect direction and intensity of light to complex organs that form images. Light detectors all contain photoreceptors, which are cells that contain light-absorbing pigment molecules. Most invertebrates have a light-detecting organ. One of the simplest light-detecting organs is that of the planarians, where a pair of ocelli called eye spots are located near the head. These allow planarians to move away from light and seek shaded locations. I do have a very nice link waiting for you, which I cannot share with you in real time, so please do click on it, watch it, and pause this video right here, come back and restart the video, and we'll continue on. So here is the planaria, and it doesn't like light. It likes to move away from light. So these are the little eye spots that you see, or the ocelli. And actually, if you looked at the ocelli, they're not all that simplified. They have these rows of cells which contain pigment. And the pigment is screening pigment that will stimulate the visual pigment in these nervous cells that have hair at the bottom, which will then take the message to the brain, the primitive brain, and then the animal moves away. Compound eyes. These are interesting because they're fascinating. Insects and crustaceans have compound eyes, which consist of up to several thousand light detectors called ommatidia. Compound eyes are very effective at detecting movement, so that's why it's so difficult to kill a fly, because it has an eye pointing in every direction and it can see the newspaper coming on it. So I do have another link, and again, please pause the video, check the link out, and come back to me. Here is the ommatidio of a fly. So here's a fly's eye. So here's one eye, and that's the other eye. But if you look at the little dots in between, that's this. And these are actually axons that are going to the fly's brain. And if you looked at one of these, there actually is a cornea, just like our eye. And there's actually a lens underneath it, which is a crystalline cone. And then there's actually a photoreceptor cell here and a rhabdom here, which will take the message to the fly's brain, which is so cool. And that's how our eyes are based on that. A single lens eye are found in some jellies, polychaetes, spiders. Spiders have single lens eyes, and many mollusks. They work on a camera-like principle. The iris changes the diameter of the pupil to control how much light enters, and the eyes of all vertebrates have single lens. So even though these are primitive, you know, these guys have eyes like us. So let's look at the vertebrate visual system. In the vertebrates, the eye detects color and light, but the brain assembles the information and perceives the image. So there's perception. You get the stimulus first, you collect the stimulus, and then you perceive it or you understand it, you integrate the signal, and you perceive it in the brain as a certain image. At least 30% of the cerebral cortex in dozens of integrating centers is active in creating visual perceptions, and we saw that. We saw that a lot of real estate in the cerebrum is devoted to vision. Transduction of visual information to the nervous system begins when light induces the conversion of cis-retinol to trans-retinol. So those cis bonds are back again. Here we are. Here's retinol in the cis isomer form, and light will actually act on it, and a bunch of enzymes will help it, and it will become the trans-retinol. So this cis bond is going to go away and it'll become straight. Enzymes will actually make it go back to the cis isomer form. Transretinol activates rhodopsin, which is a protein, which activates a G protein, eventually leading to the hydrolysis of cyclic GMP. When cyclic GMP breaks down, so GMP is the same as ATP, a MP, a DP, and M is monophosphate, G is guano, so we know that. When cyclic GMP breaks down, sodium channels will close. And that's going to hyperpolarize the cell, and the signal transduction pathway usually shuts off again as cis enzymes convert the retinol back into the cis form. So this is what happens to us. So here's our eye. And we have a cornea, and then we have an iris, which is this part. Then the iris is controlled by some muscles, which control how much light is going to hit the lens. So the pupil gets bigger or smaller depending on how big the iris gets or how small it gets. And the lens will take the signal, which is light, and it will pass through this thick, almost jelly-like material called vitreous humor, and it will fall on this yellow stuff, which you see over here, which is the lining of the inside of the eye, or the retina. So the retina is the yellow part. Underneath is the choroid, which is all blood vessels. And if you look at the part that is particularly picked up for you, it is magnified over here. So let's look at the retina. So here is the signal coming in, the light is coming in, and you would think that the light cells are going to be right there, but they're not. The first thing the signal encounters is the nerve, which is sitting in front. But these nerves and these neurons are going to be in front of the signal, and the receptors are behind the nerves. So the photoreceptors are the last layer, and there are two types. They're either rods, which look like this, or cones, which look like this. And they sit on some epithelial cells, and this is how we actually see. So let's go over the rods and the cones. So this is a rod, and that is a cone. So one is responsible for seeing in dark, and one sees color. And if you actually looked at them in real life, the rods do look like rods, and they're all these long white finger-like things, whereas the cones actually look like little cones. And they're shaped different and they're colored different too. So let's look at them in a little bit more detail, and let's see where are they in the retina. So we have an awful lot of cones. If you look at this, the number of photoreceptors, so the number of cones increases as you get to the fovea, which is the part that sees the most. And the number of rods, which is in a dashed format, increases as you get away from the fovea. So the rods are more numerous in the rest of the eye, but at the actual spot where you can see the brightest, which is the fovea, you have an awful lot of cones. And what happens inside? Well, it's still rhodopsin. So inside one of these rods and cones, what you see are these big discs. And we saw something like this when we were looking at photosynthesis. And again, this is the same idea, but this time we're using light to look at something and not to generate food. What we're doing is we're going to change the pigment or the retinol and the opsin, and we're going to move it and change it in rhodopsin. So let's look at how does it actually work. So it's again just a matter of an action potential. So we have light falling on an inactive rhodopsin molecule. The rhodopsin is going to get activated. The signal is going to be transduced by an enzyme, and this is inside the disc, and then it's going to go into the cytoplasm, where sodium ions are going to rush on in, and you get hyperpolarization. So that will tell us that there's actually light. So processing of visual information begins in the retina. In the dark, the rods and cones release neurotransmitter glutamate into synapses with neurons called bipolar cells. But in the light, the rods and cones hyperpolarize, and they shut off the release of glutamate. So the bipolar cells respond to glutamate by either hyperpolarizing or depolarizing. So on the left-hand side of the screen, you see it grayed out, and those are all the responses in dark. And let's go through them one by one before we turn to the right-hand side. In the dark, the rhodopsin is inactive. All right, the sodium channels are open. All right, and then the rod is depolarized. Yes, glutamate is received, and there's a little bipolar cell sitting around, and that can be either depolarized or hyperpolarized depending on how many receptors it got. But in the light, and now we're going to look at this side of the screen. In the light, the rhodopsin actually becomes active. The sodium channels close down. The rod becomes hyperpolarized. No glutamate is released. So then, the bipolar cell can either be hyperpolarized or depolarized depending on the glutamate receptors. So that depends on what's going on. Once we get that, then the image is taken by the nerves, the signal is taken by the nerves, and the image is processed in our brain. There are three other types of neurons that contribute to information processing in the retina. There are ganglion cells, which transmit signals from bipolar cells to the brain, and they're horizontal and amacrine cells that help integrate the visual information before it's sent to the brain. So this is 1, 2, 3. Interaction among different cells results in lateral inhibition, which will enhance the contrast in the image. So once this is integrated by the horizontal and amacrine cells, we get to see something that is clear instead of blurry. The optic nerves meet at the optic chiasm near the cerebral cortex. Sensations from the left visual field of both eyes are transmitted to the right side of the brain. Sensations from the right visual field are transmitted to the left side of the brain. Most ganglion cell axons lead to the lateral geniculate nuclei, which is in the brain and is responsible for integrating visual information. The lateral geniculate nuclei relay information to the primary visual cortex in the cerebrum. And here's what we're talking about. So what we have is an image here, and our right eye actually can see the right visual field. Yeah, it goes all the way here, but it also looks at the left visual field. Okay, and that information is transmitted here. And if you follow the blue line of the left visual field, you'll see that there are two of them. One of them goes straight to the lateral geniculate nuclei, and the other one for the left visual field also goes there. But what you also see is that in this right eye, there's a right visual field, and the information is sent not to that side, but to the opposite side, to the other lateral nuclei. So it's crossed, and so the opposite happens in the left eye. So the information is now crossed from both eyes, and that's why these nuclei have to integrate the information to make sense of what are we seeing, and then they send the information on to the primary visual cortex so we can think of it as an image. Color vision. Among vertebrates, most fish, amphibians, and reptiles, and birds, um, they have a lot of good color visions. So we believe that fish, amphibians, reptiles, and birds have very good color vision. Humans and other primates are among the minority of mammals with the ability to see color well. Mammals generally don't see color, which is why we sometimes say that our dogs probably can't tell what color it has. But that's just very interesting how some of the mammals decided not to spend energy on color and the others did. Mammals that are nocturnal usually have a high proportion of rods in the retina, so they wouldn't really see in color because they don't really need to. It's dark already. They just need to see gradations of gray. In humans, perception of color is based on 3 types of cones, each with a different visual pigment: red, green, or blue. These pigments are called photopsins, and they're formed when retinol binds to 3 distinct opsin proteins. Abnormal color vision results from alterations in the genes for one or more of the photopsin proteins. The brain processes the visual information and controls what information is captured. Focusing occurs by changing the shape of the lens, and the fovea is the center of the visual field, and it contains absolutely no rods, but a really high density of cones. And then humans have vision problems which can be corrected, but they're all because these ciliary muscles lose their ability to contract and relax. So if you have near vision, what will happen is that the ciliary muscles are supposed to contract, and these ligaments that suspend the lens are supposed to relax, and the lens becomes thicker, and you're able to focus at things that are nearby. But distance vision occurs when these muscles are relaxing and the ligaments pull the lens, so the lens becomes flatter, and so the the rays of light coming from the image, they become flatter, or they focus again. They're able to focus on distant objects. On to the next senses, gustation and olfaction. So, in terrestrial animals, gustation, which is taste, is dependent on the detection of chemicals called tastants. Olfaction, which is smell, is dependent on the detection of odorant molecules. In aquatic animals, there is no distinction between taste and smell because they can't really smell because they're already in water. Taste receptors of insects are in sensory hairs called sensilla, located on feet and in mouth parts, so they taste with their feet. In humans, receptor cells for taste are modified epithelial cells organized into taste buds. There are 5 taste perceptions: sweet, sour, salty, bitter, and umami. Any individual taste cell expresses one receptor type and detects one of the 5 tastes. One individual taste cell will express one receptor type, and it will detect one of the 5 tastes. Taste cells are modified epithelial cells organized into a taste bud, and these taste buds are located in several areas of the tongue and the mouth. Any region with taste buds can detect any of the 5 types of taste. So if you've heard that you can only taste sweet things from the tip of your tongue, that's incorrect. And here's a picture showing you how. So that would be your tongue, and it is covered by papillae. So there are little papillae in front, giant papillae in the back, and each papilla has actually a taste bud. So here is a taste bud shown here, and when we look at the taste bud, inside are these there's a little hole called the taste pore, and then there's these different cells which can detect different sensations. So the purple will taste sweet, the blue will taste salty, the yellow will taste the sour taste, and there's a green which will taste bitter, and the fifth is umami, which is this one. So there are all 5 of them which are lined up inside a taste bud, and there's a sensory neuron attached which will take the message to the brain. Olfaction. Olfactory receptor cells in our neurons that line the upper portion of the nasal cavity. So this is when you smell. Binding of odorant molecules to receptors triggers a signal transduction pathway, sending action potentials to the brain. Humans can distinguish thousands of different odors. That's awesome. Although receptors and brain pathways for taste and smell are independent, these 2 senses do interact. And here are odorant molecules going up your nose, and they actually are detected by the brain almost immediately. So, which is a good thing because you want to be away from the bad stuff. And if you really looked at it, this area is magnified. So what you see is, so these are that's your bone, and what you have are little hairs, which are going to sense the odorant molecules coming in. These hairs are attached to the chemoreceptors. They fire, and they will send messages to the olfactory bulb, and we process it as information. Well, that's the end of the nervous system and all our senses, and I hope this was helpful. I'll see you next time.